Welcome to the deep dive. Today, we're really kicking off a foundational journey into biology. Think atoms, cells, energy,
Mm-hmm.
the absolute basics that make everything tick.
That's right.
Our mission really is to connect these dots,
Yeah.
show how chemistry, like basic chemistry, underpins everything in biology. It's kind of a shortcut to understanding why things are connected, how life actually works.
It really is. And, it's fascinating because every single thing we see in biology, a thought, a heartbeat, you name it, it all traces back to these chemical and cellular rules.
Mm.
Understanding this foundation, this why, it's how we make sense of everything else from like medicine to ecology. It's all built on this surprisingly unified set of principles.
Okay, so let's start right at the beginning.
Yeah.
Matter. What are we actually made of?
Fundamentally elements, chemical elements.
Like humans need about 25 essential ones, don't they?
Yeah.
But the really cool part is how they combine. You take two sort of dangerous things,
Yeah.
like sodium explodes in water, right?
Right.
And chlorine gas, which is poisonous.
Mm-hmm.
Put them together and you get table salt,
Uh-huh.
something you can eat. These emergent properties just pop up.
Exactly. New properties emerged that weren't there in the individual parts. It's a core concept.
So what gives these elements their, personalities? Why does sodium act like sodium?
That comes down to the atom, the structure inside.
Yeah.
You've got protons, neutrons, and the really key players, the electrons.
The electrons.
Especially the ones in the outermost valence shells. They're the ones doing the interacting, determining how reactive an atom is.
Okay, so they interact. How do they actually, stick together to make stuff?
Through chemical bonds, that's the glue. You've got strong ones, like covalent bonds, where atoms share electrons.
Sharing. Okay.
And ionic bonds, where one atom actually transfers an electron to another, creating charged ions that attract like tiny magnets.
Gotcha.
But interestingly, it's often the weaker interactions, like hydrogen bonds, that are super important collectively.
Weaker but important. How does that work? Like the gecko thing.
Precisely. The gecko climbing a wall that's not one strong bond. It's millions and millions of tiny, temporary, weak interactions between its foot hairs and the surface. Add them all up, and it's incredibly strong.
Wow. So lots of small forces make a big difference.
Absolutely. And that brings us to, arguably, the most critical molecule for life, which relies heavily on these weak bonds. Water.
Ah, water.
Yeah.
H2O. Simple, but not so simple.
Not simple at all in its effects. Its unique properties all stem from its shape, its polarity, and the hydrogen bonds that form between water molecules.
Like how it sticks together. Cohesion.
Exactly, cohesion. That
Wow.
creates surface tension. You mentioned the cat drinking. That's a perfect example. The water column gets pulled up by cohesion.
That's wild.
And water moderates temperature incredibly well because it can absorb and release a lot of heat without changing its own temperature drastically.
Right.
Think coastal climates versus inland ones.
Keeps things stable.
Very stable. And then there's the really weird life-saving property,
Right.
ice floats.
Right. Most things get denser when they freeze, but water expands.
It does. It forms this crystalline lattice that's less dense. If ice sank, lakes and oceans would freeze solid from the bottom up. Life would be impossible.
And we see the impact now with, Arctic ice melt affecting animals.
Tragically, yes. It highlights how critical these physical properties are. Plus, water's polarity makes it the solvent of life. It dissolves so many things, creating the watery solutions where life's chemistry happens.
Okay, so we've got the basic elements, bonding, and the importance of water.
Right.
Where do we go next? The star player,
Yes, let's talk carbon.
carbon, the backbone of life.
Absolutely. Its magic is in having 4 valence electrons. That means it can form 4 covalent bonds. It's like the ultimate connector piece in molecular Lego.
So it can build complex shapes.
Incredibly diverse shapes, long chains, branched structures, rings. These form the skeletons of organic molecules. And you can attach different functional groups to these skeletons.
Functional groups, what do they do?
They're specific clusters of atoms that give the molecule particular properties. They can make it reactive or water-soluble or acidic.
Ah, so you tweak the groups, you tweak the molecule's function.
Precisely. Think about testosterone and estradiol, the main male and female sex hormones. They look really similar structurally, just differing by a few functional groups.
But they have vastly different effects on the body.
Exactly. Tiny chemical changes, huge biological consequences. It's fundamental.
So these carbon skeletons with their functional groups, they build the really big molecules, right? The macromolecules.
That's the next step up. These huge molecules are mostly polymers, which are long chains made by linking together smaller repeating units called monomers.
Like beads on a string.
Kind of, yeah. Cells build them using dehydration reactions, removing a water molecule to link monomers, and they break them down using hydrolysis, adding water to break the bond. That's basically digestion.
Okay. And there are four main types of these big molecules.
Four major classes, yes.
Yeah.
First, carbohydrates, sugars and starches. They're primarily fuel. Glucose is instant energy, but they can also be structural, like cellulose and plant cell walls.
Which we eat as fiber.
Right.
Yeah.
Can't digest it, but it's important. Second, lipids. Fats, oils, waxes. The defining feature is they're hydrophobic. They don't mix with water.
Why are they important?
Well, fats are great for long-term energy storage. Phospholipids are absolutely essential. They form the structure of all cell membranes. And steroids, like cholesterol, are lipids too. Cholesterol is actually a precursor for making other steroid hormones.
Okay, carbs and lipids. What's next? The really busy ones.
The workhorses,
Huh.
proteins, they do almost everything in the cell.
Like what?
Oh gosh, speeding up reactions as enzymes, transporting substances, fighting infections with antibodies, enabling movement in muscles, providing structural support like keratin in hair or collagen in skin. The list is huge.
And they're all made from just 20 building blocks.
Just 20 different amino acid monomers, yes, linked together by peptide bonds into long chains called polypeptides. But the sequence is just the start.
What do you mean?
The crucial thing for a protein is its specific, intricate, three-dimensional shape. That shape determines its function. It has to fold just right.
So shape equals function.
Absolutely. Think of a key fitting into a lock. An enzyme's active site has to perfectly fit its substrate. Opiates work because their shape mimics natural signaling molecules, fitting into receptors in the brain.
And if the shape gets messed up?
Then the protein stops working. That's called denaturation. Things like extreme heat or changes in pH can cause a protein to unravel.
Like cooking an egg white, it goes solid.
Exactly. The proteins denature and tangle up. And that's why a high fever is dangerous. It can denature critical proteins in your body.
Wow. And even tiny changes matter. You mentioned sickle cell disease.
A devastating example. Just one amino acid change out of hundreds in the hemoglobin protein changes its shape enough to cause red blood cells to become sickle-shaped, leading to all sorts of problems. It really underscores how critical that precise 3D structure is.
Incredible. Okay, so proteins do the work. What's the last group? The info storage.
The information carriers, nucleic acids, DNA and RNA.
The famous double helix.
That's DNA. It stores the genetic blueprint. RNA is involved in actually using that information, taking the instructions from DNA to build proteins. This whole process is called gene expression.
And DNA structure is key here too.
Absolutely. The double helix, with its two strands running in opposite directions anti-parallel, and the specific way the bases pair up, A with T, G with C, that's how information is stored accurately and copied faithfully generation after generation.
Understanding this structure must have revolutionized biology.
Immensely. It opened the door to genomics, studying entire sets of genes. By comparing genomes, say, between humans and chimps, or even humans and frogs, we can see evolutionary relationships written in the DNA sequences. It's powerful evidence.
Okay, so we've got the molecules. Now let's assemble them into the fundamental unit of life itself. The cell.
The cell. Everything comes back to the cell. Cell theory basically says all living things are made of cells, and the cell is the smallest thing that can be considered truly alive.
And there are different basic types.
Two main types. Prokaryotic cells, simpler, smaller, no nucleus, think bacteria. And eukaryotic cells, larger, more complex, with a nucleus and lots of internal compartments called organelles. That's us, plants, fungi, animals.
Why the compartments in eukaryotic cells?
It's all about efficiency and specialization, like having different rooms in a house for different activities. This internal compartmentalization allows eukaryotic cells to be much larger and more complex.
So let's take a quick tour inside a eukaryotic cell, the control center first.
That's the nucleus. It houses the cell's DNA, packaged up with proteins into chromatin. It's where the genetic instructions are kept safe and read out.
And protein factories.
Ribosomes. They read the RNA messages coming from the nucleus and assemble amino acids into proteins. Some float free, making proteins used within the cell. Others are attached to a network.
The endomembrane system. That sounds complex.
It is. It's like the cell's internal manufacturing and shipping network. It includes the endoplasmic reticulum, or ER. The rough ER has ribosomes and makes proteins destined for secretion or membranes. The smooth ER makes lipids, detoxifies drugs.
Like in liver cells.
Exactly. And it stores calcium, which is crucial for muscle contraction. Then there's the Golgi apparatus. Think of it as the post office.
Sorting and packaging.
Yep. modifying proteins and lipids from the ER, sorting them, and packaging them into vesicles for delivery elsewhere in the cell or outside. And lysosomes are the recycling centers, containing digestive enzymes to break down waste or engulfed material.
A whole internal logistics system. Amazing. What about energy?
The power plants, mitochondria and chloroplasts.
Chloroplasts only in plants and some algae, right, for photosynthesis.
Correct. Mitochondria are in nearly all eukaryotic cells, and they're where cellular respiration happens, breaking down fuel molecules like glucose to generate ATP, the cell's energy currency.
And they have a weird origin story, the endosymbiont theory.
Fascinating theory, and lots of evidence supports it. It suggests mitochondria and chloroplasts were once free-living prokaryotes that got engulfed by larger cells and formed a symbiotic relationship. They even have their own DNA and ribosomes, similar to bacteria.
Wow, and holding it all together, giving it shape.
That's the cytoskeleton, a dynamic network of protein fibers, microtubules, microfilaments, intermediate filaments. It provides structural support, helps the cell move, moves organelles around. It's the cell's internal scaffolding and highway system.
Cells aren't just floating around, though, are they? They interact.
Definitely not in isolation. Plant cells have rigid cell walls outside their membrane for support. Animal cells have an extracellular matrix, or ECM, a complex mesh of proteins like collagen outside the cell.
Does that matrix just sit there?
Oh no, it's active. Cells connect to it via proteins called integrins, which span the membrane and link to the cytoskeleton inside. This connection allows the ECM to influence cell behavior, even gene activity.
And cells connect directly to each other.
They do. Plants have plasmodesmata, channels through their walls. Animal cells have different types. Tight junctions seal cells together, desmosomes anchor them, and gap junctions are communicating channels, like plasmosmata, allowing small molecules to pass between cells.
So it's all interconnected inside and out.
It's a beautiful integration. Think of a white blood cell engulfing a bacterium. Cytoskeleton helps it move, membrane changes shape. Lysosomes provide digestive enzymes made by ribosomes and processed by the ER and Golgi, all powered by ATP from mitochondria. It's a coordinated symphony.
Let's zoom in on that boundary, the cell membrane. You called it a fluid mosaic.
Yes, the fluid mosaic model. It emphasizes two key things. It's fluid, meaning the components can move around laterally, and it's a mosaic of different molecules, mainly phospholipids and proteins.
Phospholipids. They have the split personality, right?
Mm.
Water-loving head, water-fearing tails.
Exactly, they're amphipathic. This causes them to spontaneously form a stable bilayer in water, with the tails hiding inside and the heads facing out. That's the basic fabric of the membrane. Cholesterol fits in there too, helping regulate the fluidity.
And this structure makes it picky about what gets through. Selective permeability.
Precisely. Small, non-polar molecules like oxygen can slip through easily. But charged ions and larger polar molecules like glucose need help from transport proteins embedded in the membrane.
How does that transport work? Is it always passive?
Not always. Passive transport doesn't require the cell to expend energy. Things move down their concentration gradient from high concentration to low. Simple diffusion is one way.
Spreading out.
Yep. Then there's osmosis, which is specifically the diffusion of water across a selectively permeable membrane. This is crucial. Think about how cells react in different solutions, swelling or shrinking based on water movement or tonicity.
Right. And if things need help crossing, even passively.
That's facilitated diffusion. It still follows the concentration gradient, so no energy needed, but it uses protein helpers. Channel proteins form tunnels, like aquaporins, just for water. Carrier proteins change shape to shuttle specific molecules across.
Okay, so that's downhill. What if the cell needs to move something against the gradient uphill?
Now you need energy. That's active transport. The cell has to spend ATP to pump substances where they were already more concentrated.
Like the sodium-potassium pump. I remember hearing about that in nerves.
The classic example. It constantly pumps sodium out and potassium in, maintaining crucial ion gradients that are essential for nerve signals and many other processes. It uses a lot of the cell's energy.
And what about really big stuff, like engulfing a bacterium?
That requires bulk transport. Endocytosis is bringing stuff into the cell. Phagocytosis, cell eating, is engulfing large particles. Penocytosis, cell drinking, is taking in droplets of fluid. There's also receptor-mediated endocytosis, which is very specific, using receptors to grab particular molecules.
Like cholesterol uptake, you mentioned a disease.
Familial hypercholesterolemia, yes. A defect in the LDL cholesterol receptors means cells can't take up cholesterol properly, leading it to build up dangerously in the blood. It shows how vital these transport systems are. And exocytosis is the reverse vesicles fusing with the membrane to release substances out of the cell.
So cells move things in and out,
Mm.
but they also need to communicate, coordinate actions.
Absolutely critical, especially in multicellular organisms. Cell communication can be short range, like cells touching or releasing local signals, or long range, using hormones that travel through the bloodstream.
How does a cell actually receive and respond to a signal?
It's typically a three-stage process called a signal transduction pathway. First is reception. A signaling molecule, a ligand, binds to a specific receptor protein, usually on the cell surface.
Like a key fitting a lock again.
Exactly. That binding triggers the second stage, transduction. The signal is relayed, often through a cascade of changes inside the cell, think dominoes falling, often involves phosphorylation cascades, where proteins called kinases activate each other by adding phosphate groups, like molecular switches.
Perfect analogy. And sometimes second messengers like cyclic AMP or calcium ions are involved, rapidly spreading the signal throughout the cytoplasm.
And the final step.
Response. The cell actually does something, maybe activates a gene in the nucleus, changes the activity of an enzyme, opens an ion channel. The specific response depends on the signal and the cell type.
Like adrenaline making liver cells release glucose.
Precisely. And what's amazing is the amplification. One hormone molecule binding to one receptor can trigger a response involving millions of molecules inside the cell. It's incredibly efficient.
Okay, this cellular machinery is mind-blowing. But what powers it all? Where does the energy come from?
Metabolism.
Hmm.
That's the sum total of all the chemical reactions happening in an organism. It's all about managing energy.
Energy. Kinetic potential.
Right. And the fundamental rules are the laws of thermodynamics. First law. Energy can be transferred and transformed, but not created or destroyed. Conservation of energy.
Second law. Every energy transfer increases the entropy or disorder of the universe. Basically, some energy always becomes unusable, often lost as heat. Living things seem orderly, but they do it by increasing disorder elsewhere.
So how do cells manage energy in reactions? Which ones happen spontaneously?
That's determined by the change in free energy, symbolized as Runge. Reactions with a negative Yi are exergonic. They release energy and can happen spontaneously. Reactions with a positive Yi are endergonic. They require energy input.
Like rolling uphill versus downhill.
Exactly. And cells need to run lots of uphill endergonic reactions to build things. How do they do it? By coupling them with exergonic reactions.
Using an energy currency.
Precisely. The main energy currency is ATP adenosine triphosphate. Breaking the bond of its last phosphate group releases a nice packet of energy.
So ATP breakdown powers the uphill reactions.
Yes, through energy coupling. The energy released from ATP hydrolysis is used to drive the endergonic reaction, often by temporarily transferring the phosphate group to one of the reactants, making it more reactive.
And the cell is constantly remaking ATP.
Constantly. The ATP cycle is relentless. Energy released from breaking down food, catabolism, is used to stick the phosphate back onto ADP, regenerating ATP. Then that ATP is used to power cellular work, anabolism, transport, movement. It's estimated a working cell recycles its entire pool of ATP molecules roughly every minute.
Wow. Okay, so reactions happen if they release energy, or if they're powered by ATP. But are they fast enough?
Often no. Even spontaneous reactions might happen incredibly slowly on their own. They need a push to get started, an initial energy investment called the activation energy barrier.
Like needing a spark to light gasoline.
Perfect analogy. That spark is the activation energy. In cells, enzymes act as biological catalysts. They dramatically lower that activation energy barrier.
So they make reactions happen much faster.
Millions, even billions of times faster. Crucially, they do this without being used up in the reaction and without changing the overall energy release. They just make it easier for the reaction to happen.
And they're specific.
Highly specific. Each enzyme typically catalyzes only one or a few specific reactions because its active site has a unique shape that fits only its particular substrate. Like a lock and key, or maybe more like a glove fitting a hand-induced fit.
And conditions matter. Temperature, pH.
Absolutely. Each enzyme has optimal conditions where it works best. Too hot, too cold, too acidic, too alkaline, and its shape can change, reducing its activity or even denaturing it completely.
Like pepsin in the stomach needing high acidity.
Exactly. Cells also regulate enzymes, turning them on or off as needed. One common way is feedback inhibition, where the final product of a metabolic pathway inhibits an enzyme early in that same pathway. It prevents the cell from making too much of something it doesn't need.
Efficient control. Okay, let's connect this to the big energy processes. How do we get energy from food?
That comes down to redox reactions, oxidation, and reduction. Remember oil rig. Oxidation is loss of electrons.
Hmm.
Reduction is gain of electrons.
Hmm.
In processes like cellular respiration, electrons are stripped from glucose. Glucose is oxidized and eventually transferred to oxygen. Oxygen is reduced, forming water. As electrons move from glucose to oxygen, they go to a more stable, lower energy state, releasing energy along the way.
Like rolling downhill energetically.
Precisely. But the cell doesn't let them fall all at once. That would be like an explosion. Instead, it uses an electron transport chain. Electrons are passed down a series of carrier molecules, releasing energy in small, manageable steps.
Where does this happen?
Mostly inside the mitochondria. It starts with glycolysis in the cytoplasm, splitting glucose into pyruvate. Then pyruvate enters the mitochondrion, gets converted, and feeds into the citric acid cycle, which breaks it down further, releasing CO2 and capturing high-energy electrons onto carriers like NADH.
And then the electron transport chain.
Yes. That chain uses the energy from those electrons to pump protons, H+ ions, across the inner mitochondrial membrane, creating a steep gradient of the proton motive force.
Like building up water behind a dam.
Good analogy. Then, in chemiosmosis, those protons flow back across the membrane through a molecular machine called ATP synthase, and the flue drives the synthesis of ATP. Lots of ATP. This final stage, oxidative phosphorylation, generates the vast majority of ATP from respiration.
What if there's no oxygen?
Then cells can use fermentation. Glycolysis still happens, making a little ATP, but instead of sending electrons down the transport chain to oxygen, they're passed to organic molecules to regenerate the carriers needed for glycolysis to continue.
Like making alcohol in yeast or lactic acid in mussels.
Exactly. Alcohol fermentation produces ethanol and CO2. Lactic acid fermentation produces lactate. It's much less efficient at making ATP than aerobic respiration, but it keeps the cell going when oxygen is scarce.
So respiration breaks down glucose. Where does the glucose come from in the first place?
For most ecosystems, it comes from photosynthesis. This is the process plants, algae, and some bacteria use to capture light energy and convert it into chemical energy stored in glucose.
Using CO2 and water.
Right. The overall equation is kind of the reverse of respiration. Light energy plus CO2 plus water glucose plus oxygen. Importantly, the oxygen released comes from the splitting of water molecules, not from CO2.
Where does this happen in plants?
In the chloroplasts, it has two main stages. The light reactions happen in the thylakoid membranes. Pigments like chlorophyll capture light energy, which excites electrons. These electrons flow down another electron transport chain, generating ATP and electron carriers, NADPH, and splitting water releases oxygen.
So light energy gets converted to chemical energy in ATP and NADPH. Then what?
Then the Calvin cycle takes over in the stroma, the fluid inside the chloroplast. It uses the ATC and NADPH from the light reactions to fix carbon from CO2, incorporating it into organic molecules and then reducing it to form sugar, glucose.
It's like respiration and photosynthesis are two sides of the same coin, cycling energy and matter.
Beautifully put. They are fundamentally linked, driving the flow of energy through almost all life on Earth.
Okay, one last fundamental process.
Yeah.
How do cells make more cells? How does life continue?
Through cell division. It's essential for reproduction in single-celled organisms, for development from a fertilized egg into a multicellular organism, and for growth, repair, and renewal throughout life.
Making sure the genetic information gets passed on correctly.
Absolutely. The cell's DNA is organized into chromosomes. Before dividing, the cell meticulously duplicates all its chromosomes. Then, during division, it ensures that each new daughter cell gets one complete identical set.
This happens in the cell cycle.
Yes, the cell cycle is the whole sequence. Most of the time, the cell is in interphase growing, replicating its DNA in the S phase, and preparing for division. Then comes the M phase, which includes mitosis, the division of the nucleus and chromosomes, and cytokinesis, the division of the cytoplasm.
Mitosis itself has stages, right? Prophase, metaphase.
Yes, a series of carefully orchestrated steps. Chromosomes condense, the mitotic spindle made of microtubules forms to capture them. They all line up perfectly in the middle at the metaphase plate, and then the duplicated sister chromatins are pulled apart to opposite poles of the cell.
Ensuring each new nucleus gets a full set.
Exactly. Then cytokinesis splits the cytoplasm, forming two distinct daughter cells, each genetically identical to the parent cell.
But this cycle needs control, doesn't it? Cells can't just divide whenever.
Definitely not. There's a sophisticated molecular control system involving proteins called cyclins and enzymes called kinases, specifically CDKs. They act at critical checkpoints like tool booths, ensuring everything is ready before the cell proceeds to the next stage. If conditions aren't right, the cycle pauses.
What happens if that control breaks down?
That's essentially what cancer is. Cancer cells are cells that have escaped the normal cell cycle controls and divide uncontrollably. They often ignore signals like density-dependent inhibition, stop dividing when crowded, or anchorage dependence, only divide when attached.
Leading to tumors.
Exactly. And potentially metastasis, where cancer cells spread to other parts of the body. It's a devastating consequence of losing that fundamental cellular regulation.
So we've gone from atoms and water chemistry through macromolecules and organelles, energy flow, all the way to cell division and its control. It's quite a journey.
It really covers the absolute bedrock of biology.
It feels like the key theme is this incredible organization. From simple parts building complex structures, and these structures having amazing emergent functions.
That's a perfect summary. Life is this hierarchical system, constantly managing energy, matter, and information flow. It's a dynamic balance built on these fundamental chemical and cellular rules.
And understanding these rules is the key to understanding, well, everything else in biology.
It truly is, and it makes you wonder, what happens when these building blocks interact in ways we haven't even thought of yet? What new properties, what new understanding might emerge? That's the exciting frontier.
A lot to think about. Keep exploring, keep questioning, and keep diving deep. Thanks for joining us on this deep dive.